Otherwise, I'll just go and restart this explanation. All right, so I was explaining this, depth electrode, which consists of the microwire, and that's inserted into the brain like this way. And then from the tip of this microelectrode, you can record the spiking activity. This is over the 10 seconds here. So you can see, it's called a high pass filtering signal, but you don't need to understand this. But this almost like a raw data from you can see sometimes there's this kind of voltage fluctuation that extends really sharply. And then if you focally analyze this using some kind of machine learning technique nowadays to sort of detect and categorize the shape of this thing in a more refined time window over, this is roughly like 1 millisecond or something like that. It's very, very short duration of this thing, so you need to magnify this quite a bit, and you need to record it sample at a very, very high sampling frequency. And then depending on the let's say if the neurons are located like this way, and then the spike neuron bodies like this, and then if the electrodes happen to be close to this cell body, then it might be like generating a large amplitude spike like here. But if it's a bit farther, then the spike may be smaller like this way. And depending on the type of the ion channels and so on, very complicated story, depending whether it's closer to axon or dendrite, the shape also changes like here. And so because of this type of the biophysical mechanism, based on the shape of the spikes, we can confidently say that it is coming from one neuron or the other. And then when the people present the various kind of images to the same patient whose data was recorded like this way. In this particular case, it may be a fictitious case, I don't know about this, but when they look at the response of this single tick of the spikes are counted as each of this vertical tick mark like here or here, and then align it with multiple trials. Let's say this is our first trial of the spider, second, third, fourth, fifth, and the spider trial, and I presented from this duration to this duration, okay, this to this time. Then after seeing the spider, this neuron fired a lot like this. After seeing this monkey, fired a lot as well. This is a squirrel and something like that, and tiger and so on. By the way, this is a kind of a neuron that we published together. Although I wasn't involved in this recording part, but more on the fMRI side of this. And this particular paper reported this type of the neuron that responded only to animals, but not others. So any kind of animals, from elephants, dogs, or dolphin or bird and things like that, and even Shrek also generate a response like this. On the other hand, this cluster 3 neuron responded, in this case, this particular instance of this, rabbit-like fluffy animal, not to the other. And then other kind of categories like a human face or the building, it didn't respond. So the left kind of that neurons are more or less like a categorical response. There are many examples of the images that can generate a response. Whereas here's a particular example, particular concept that generate this response. So far, does it make sense? Any question? Okay. And they also did a bunch of experiments. And here's one particular famous neuron that was discovered in the posterior hippocampus. And what they did was to give you more detail of this experimental situation. What they did was in the morning of this experimental session, they presented a bunch of images to the patient who are recorded with this wire, and asked them what kind of hobbies or TV show or they like and so on. And this particular patient said that she liked the Friends, the TV drama show. So the experimenters included a bunch of photos from the Friends characters, and this is a Jennifer Aniston's Rachel character, and that produced a bunch of responses like this. And then during the lunch break, this experimenter skipped lunch pretty much, and then just collected a bunch of more similar faces like Jennifer Aniston or the different example of the Jennifer Aniston. And then when they did that, they found that different views, different examples of Jennifer Aniston produced a bunch of good responses, like original Jennifer Aniston in the morning. On the other hand, other female actress didn't show much of the response here. And then other categories of the stimulus didn't produce much response as well. So they named this maybe as a Jennifer Aniston neuron. Interestingly, when they showed that it happened to be the case, but they were, when she was married with Brad Pitt, there were lots of couple photos like this. But when they are paired, then there was no much reduced response to the pair of the Jennifer Aniston and Brad Pitt. And this one is so-called Catwoman neuron or Halle Berry. And this is similar to the Jennifer Aniston example, but when this Halle Berry, the actress, was presented, this neuron responded. But also the line drawing of the Halle Berry, but also Halle Berry in the Catwoman costume. This is in the Batman movie, also responded almost the same way, but not to the other actress again, and including Mother Teresa. But also interestingly, this text of the Halle Berry also generated a huge response, and potentially even more, stronger response than the visual response. This seems to suggest that this neuron is actually responding to the concept of the Halle Berry, not the particular image of the Halle Berry. And then this is also another very interesting situation where when there's an recording opportunity from the left anterior hippocampus, then this particular person recognized Sydney Opera. So Sydney Opera was presented, there was a lots of response, and Sydney Opera as a text also, generated a huge response here. But it's also interestingly the case that Sydney Opera, like building, it's the Baha'i temple in I think Middle East, also generated almost as strong response as Sydney Opera. And afterwards, when the patient was interviewed, he or she reported that he or she thought that these two things are the same thing from the vision. But this wasn't the case for the other, building. So it's not the kind of the building category neuron, like an animal category neuron, right? And also, again, not the general response to other different kind of famous landmark or famous person type response. So this example suggests that what matters for this neuron's response is what the patient actually saw or recognized in this image or the concept. But that's called, that's the reason why it's called concept cells or meaning kind of cells. Here, the Baha'i was presented as a text, but it didn't, induce a response. So this was not recognized as Baha'i temple. And these researchers also tried a backward masking experiment with while recording these neurons. So when these right entorhinal cortical neurons were found to respond to this World Trade Center image when this image was presented for relatively long duration, 264 milliseconds. Then it generated a lots of responses. And then this blue or red thing here is a patient report that whether they saw the object clearly or not. I think this one, or whether the presentation was a face or not, or something like that. It's very simple kind of behavior response. And then when they reduce the duration into 33 milliseconds, it's very, very short right now here. Then almost no response was generated across any images. But when it starts to become like 66 milliseconds, that's roughly like 4 frames on your 60 hertz monitor. Then sometimes like 40% of the trials, this patient reported seeing this World Trade Center, and then it's accompanied with this response. And when he denied seeing it, then there was no response here. And then when it reached to 132 milliseconds, then most of the trials, he saw it as a World Trade Center, and then that corresponds to this blue response. But rarely, when he missed it, this red trials, then he didn't show that the neuron also didn't show the response. So that seems to be a strong correlate of the consciousness when the stimulus itself is fixed and then repeated across trials over the backward masking. Okay. And this is the average of other different neurons and also different stimuli. So here's a recognized trial average, and this is an unrecognized average trials. And when the duration is changed from 32 to 60 to 64 milliseconds. And as you can see, even if the duration is very brief, when image was recognized, the response is very big. But when it's not recognized, this diminishes, and then it can be shown in each of the trials. But as you go for the longer duration trials, then the response itself seems to be big, but the subject sometimes missed it. Okay, so that leads to the kind of the question about this vision, concepts, and memory, and its relationship. So here's a standard sort of the idea or model among the vision, concepts, and memory proposed by Quiroga. He is the person who has been doing this single neuron recording from the patient, mostly in the area around the hippocampus. And most of our knowledge about the response in B1 or IT or along these ventral areas come from either functional MRI in human or lesions in humans or single neuron recording in monkeys. But within the hippocampus, because the epilepsy surgery takes place a lot, we tend to have this kind of single neuron response experiment also happening in the human brain as well. So there is some kind of extrapolation about the result, but we think this is a reasonable kind of given the correspondence or convergence from lesion and fMRI and single neuron experiment. So at the earliest stage of the visual processing, people usually assume that the response is determined by simple kind of features such as orientation or stimulus and so on. And that's what I explained for the property of the response in V1. And then as V1 goes to V2, V3, V4, and so on, this response is combined in a complicated manner to eventually arrive this neuron that responds to the face. And then from there, somehow further complicated, nonlinear operation happening. And then within temporal areas, this specific response to the Diego Maradona starts to appear. And as you saw in the example, at this level, in the hippocampus or amygdala, the modality of the sensory input doesn't matter. The text of the Diego Maradona would probably evoke the response from this neuron. And also other experiment has shown that the sound of the Diego Maradona, like pronunciation of that, also evokes a strong response and so on. So the idea is that the visual experience or perception is formed through this, ventral pathway processing to arrive at the IT for recognition. And then that is related to the memory and also concept formation at the level of the hippocampus. That's the sort of the model they suggest. So far clear? No question? Okay. And then, so this is a kind of the model they propose as well. So if you start think about whether the concept is represented within a particular area of the brain, such as the hippocampus, you might start to wonder if there is any kind of limit in terms of the number of the concepts that we can acquire, right? So, for example, this is also called a problem of the grandmother neuron hypothesis. If you have any concept, conscious experience of the concept, such as Luke Skywalker or Yoda or Darth Vader or whatever, and then correspondingly, if you have a neuron that fires for each of them with completely flexible kind of sensory input, such as Luke Skywalker's image or concept or pronunciation or anything like that, then you'd need at least same number of the neurons as the number of the concepts that you have. And how many concepts do you have in the in your life? That's the question. So here I'll ask, as a sort of your guess, how many concepts do you think we have as a human? So option A is more than 200,000 concepts. More. And then option C is that less than 4,000. And then B is intermediate between these. So what can you type your guess? Wait a sec. Okay, so which one is your guess in terms of number of the concepts that we have as a human? One is more than 200,000. And by the way, I should also say that the number of the neurons, entire, neurons in the brain is 10 to the 8, right? So that means that the 0, 0, 0, 0, 0, 0, 0, 0. That's the number of the neurons, cortical neurons in the brain. And also, this is just a tiny part of the brain, hippocampus. So it's likely to be very, very small. But anyway, okay, so everybody seems to be happy with this option. More than 200,000 concepts. So the number here is roughly the same as, I think, the number of the words entry in the Oxford or Merriam-Webster dictionary in English. And the number of the 4,000 is roughly the number of the faces that we can distinguish in everyday life. So nobody really knows how many distinct concepts we have. But the kind of the concept that I'm talking about here, like a typical concept such as Luke Skywalker with different kind of, views, or Yoda or Darth Vader and so on. If you start thinking about that, then because the number of the even words are relatively limited, less than a million, probably the number of the concepts that we can think about like this way, Luke Skywalker or Yoda or something like that, is intermediate, probably around here. But I don't know whether I'm wrong or not. But that's one way to think about this issue. Is there any opinion about this or question about this? Because not everybody probably agreed with my guess. Okay. Maybe you can type your guess in the chat during the next rest period. But anyway, so in terms of the psychological theories of concepts, the most famous one is this Aristotelian categories of concepts. So Aristotle tried to categorize or enumerate all the objects, in the world that can be recognized and then classified it into the, ways. And that roughly the approach to this, dictionary kind of concept, right? And then he would say maybe much less than 200,000. Especially at that time, there was no such concept like a TV or internet or Ethernet or whatever, right? And then there was a significant expansion of this theory called prototype theory by Eleanor Roche around 1973. And what Eleanor Roche proposed was that no, category is not something like this rigid thing like Aristotle has been proposing. It's more like there is a prototype of a concept, such as dog here, and then the exemplar, one of the sort of peripherally oriented kind of thing that is far from this typical thing. And there is a grade of this prototype or typicality of the category. That's what she claimed as a prototype theory of the concepts. And this has been quite influential in the theory of the categorization or conceptualization or whatsoever right now. So at this stage, I'll just take a break, and then we'll come to the last part of the today's topic, which is probably most controversial at this stage. Okay, 4-minute break now.